Hello and welcome back. This lecture is part 1 for this topic and will focus on energy and metabolism. Particularly, part 1 will cover metabolism and metabolic pathways, kinetic versus potential energy, the laws of thermodynamics, Gibbs free energy, equilibrium and ATP hydrolysis, and energy coupling. Part 2 of this topic will focus on enzymes and enzyme metabolism. At the end of this lecture, you will be able to explain the processes of energy transformation as they relate to cellular metabolism. Particularly, you will be able to explain the relationship between energy and work, differentiate between kinetic and potential energy, and the laws of thermodynamics, and you will learn ATP structure and explain how ATP hydrolysis is used to perform cellular work. So let's begin. Energy and metabolism. As you well know, virtually every task performed by living organisms requires energy. Organisms require energy to perform labor and exercise, but we also use considerable energy while thinking and even while we sleep. Living cells constantly use energy, but from where and in what form does this energy come from? The living cell is a miniature chemical factory where thousands of reactions occur. In animals, a complex process called cellular respiration is able to extract energy that's stored in the foods we eat, like sugars. And we'll talk all about that in the next lecture chapter. Cells apply this energy to perform work. Some organisms even convert energy to light, as in bioluminescence. Scientists use the term bioenergetics to discuss the concept of energy flow through living systems, such as cells. The image on the right shows the energy flow from the sun, which is converted by plants into chemical energy, and then animals can consume that chemical energy, which will eventually be passed on to decomposers. We see that this energetic pathway also releases energy as heat in a few places. We'll talk in depth about energy transfer and heat in this lecture. In the following chapter lectures, you'll learn about cellular respiration and photosynthesis. Cellular processes which build and break down complex molecules occur through stepwise chemical reactions. Some of these chemical reactions are spontaneous and release energy, while others require energy to proceed. Just as living things must continually consume food to replenish what they've used, cells must continually obtain more energy to replenish what the many energy-requiring chemical reactions have used. All of the chemical reactions that transpire inside the cells, including those that use and release energy, are the cell's metabolism. Metabolism is the totality of an organism's chemical reactions. Here's a map to help you visualize metabolism. Within the metabolic map lies the pathways that make or break carbs, proteins, lipids, and nucleic acids, our 4 macromolecules from Lecture 5. It shows that and more. And fortunately, or maybe unfortunately, you'll be learning some of this in this course and much more in future biochemistry and nutrition courses. This metabolism map is essentially a continual series of metabolic pathways. A metabolic pathway is a series of interconnected biochemical reactions that convert a substrate molecule or molecules step by step through a series of metabolic intermediates, eventually yielding a final product or products. They are the pathways of how we turn chemical energy from foods into energy that we can use and waste. At the simplest level, a metabolic pathway begins with a specific molecule and ends with a product. Each step is catalyzed by a specific enzyme. The video here is a 3D depiction of an enzyme. Each step of the pathway requires a specific enzyme. Reaction 1 begins with molecule A and is catalyzed to become molecule B by enzyme 1. Reaction 2, we see molecule B can be converted to molecule C with enzyme 2. And the final step of this metabolic pathway is reaction 3, where molecule C is converted to molecule D, catalyzed by enzyme 3. This is a simple metabolic pathway. Here again is the metabolism map with the metabolic pathways vital to human life. Bringing this discussion back to lecture 1 and the concept of emergent properties, metabolism is an emergent property of life which arises from orderly interactions between molecules. So let's break down metabolism. There are two arms of metabolism, two basic types, catabolism and anabolism, and they are opposites. Catabolic pathways release energy by breaking down complex molecules into simpler, smaller compounds. Catabolism literally means destructive metabolism, and to help you remember that, think catastrophe, as it has the same root word. Cellular respiration, which is the breakdown of glucose in the presence of oxygen, is an example of a pathway of catabolism. And again, we'll learn cellular respiration in the next lecture. Anabolic pathways consume energy and build larger, complex molecules from smaller, simpler ones. For example, the synthesis of a protein from amino acids is an anabolic pathway. And if you've ever heard of anabolic steroids, these are used to stimulate muscle growth and appetite in athletes and bodybuilders. Of course, the purpose of metabolism is to harness energy. As I mentioned, virtually every task performed by living organisms requires energy. But what is energy? Energy is the capacity to cause change. Very simple, right? You already know that energy exists in various forms, and you probably can list a few of them. Some of these forms of energy can perform work. Work is a term you'll see often associated with energy science, especially when you get to physics. When work is performed by a system, it just means that energy is transferred by the system to its surroundings. So energy is the capacity to cause change and/or the ability to do work. Examples of energy are electrical energy, light energy, and heat energy. I do want to point out the units. It's very important to be aware of units in science classes. We can express energy with different units. You've already learned about calories and kilocalories, but the official unit of energy of the SI system, which is the International System of Units, is capital J for joule or kJ for kilojoule. The SI system is like the modern form of the metric system. And the important thing about energy is that it can be converted from one form to another. In this image, we see the conversion between kinetic and potential energy. So, what are kinetic and potential energy? Kinetic and potential energy are the two major types of energy. Any form of energy that you can name fall within these two categories. Kinetic energy is energy associated with motion. If it's moving, it has some type of kinetic energy. Thermal energy is the kinetic energy that's associated with the random movement of atoms or molecules. In this video, we see movement. The red molecules are moving faster than the blue molecules, which tells us that the red molecules have more kinetic thermal energy. Heat is thermal energy that's transferred between objects. We see in this video that the heat from the red molecules is being transferred to the blue molecules, so they're becoming purple. And this happens until equilibrium is reached. So heat is a type of thermal energy. We know heat is related to temperature, and temperature is related to movement of molecules. Higher temperature means more kinetic energy. Examples of kinetic energy are thermal energy, vibrational energy, rotational energy, and translational energy. Notice how they all have movement. Potential energy is energy that matter possesses because of its location or structure. One form of potential energy is chemical energy. Chemical energy is potential energy available for release in a chemical reaction. This would be an example of chemical energy due to structure of the molecule. So back to our metabolic map, these pathways are releasing chemical energy stored within these molecules so that it can be harnessed to do work and keep us alive. Here are other examples of chemical energy. An octane molecule used in car fuel and triglyceride used as fuel in our own bodies. Here are examples of potential energy that are due to position. As we already know, the body works hard to create certain concentration gradients within our cells. The sodium-potassium pump is a great example. Well, the position of those molecules and that concentration gradient is a form of potential energy. If left alone, that potential energy will be transferred into kinetic energy as those molecules diffuse, move, across the membrane, as is shown here. The archer is also displaying potential energy as he has drawn the bow and arrow back, ready to release. The potential energy stored would propel this arrow forward if he released it. Other examples of potential energy are gravitational energy, elastic energy, electrical energy, and nuclear energy. So kinetic energy is the energy of motion, while potential energy is the energy of location and structure. We already mentioned that forms of energy may be converted from one to another. For example, an archer must use chemical energy from food to fuel kinetic energy to draw back the bow. This creates potential energy, which can then be released with the arrow to create more kinetic energy. These energy transformations follow certain laws of thermodynamics. Thermodynamics is the study of energy transformations. Before we get into the laws of thermodynamics, we have to understand how systems interact with their surroundings. An isolated system is unable to exchange energy or matter with its surroundings. A thermos is a perfect example. This is why when you put hot or cold liquids inside, they're able to maintain that temperature. There's no exchange of energy or heat to the surroundings. In an open system, energy and matter can be transferred between the system and its surroundings. Organisms are open systems. We are able to lose and absorb heat while interacting with our surroundings. A glass of ice-cold water is another example. Over time, the heat from the surrounding air will warm the water and melt the ice until it has reached equilibrium. So let's get into the laws of thermodynamics. We are going to cover two of them. According to the first law of thermodynamics, the energy of the universe is constant. This means energy can be transferred and transformed, but it cannot be created or destroyed. You've probably heard this law shortened to energy can neither be created nor destroyed. This law is also called the principle of conservation of energy. This law explains the scenario of the melting ice water and the archer. They exemplify transfer and transformation of energy. In the lower image, we see chemical energy from food converted to kinetic energy as a kid rides a bike. We also see light energy from the sun converted to chemical energy as plants make macromolecules. The second law of thermodynamics involves entropy. During every energy transfer or transformation, some of that energy is unusable, which means that it's not going to do work, and it's often lost as heat. According to the second law of thermodynamics, every energy transfer or transformation increases the entropy, which is capital S, of the universe. So what is entropy? Entropy is a measure of molecular disorder or randomness. It's that simple. Energy is disorder or randomness. So, because every transfer or transformation increases entropy, the universe's entropy is always increasing. This is written as delta S is greater than 0. This means the change in entropy is positive, greater than 0. That delta sign means change. So the change of entropy greater than 0, which makes it positive. We are going to focus on this law of thermodynamics and the concept of entropy a little bit more because it's important in understanding energy and metabolism. To reiterate, every energy transfer loses energy in a form that's unusable, and usually that is as heat. Living cells unavoidably convert organized forms of energy into heat, which is a more disordered form of energy. In other words, energy transfers are very inefficient. Look at the image of the cell on the right. This is a basic schematic of anabolism and catabolism, making and breaking molecules. Notice the heat that's lost at both pathways. Every reaction loses some energy as heat. The more energy that a system loses to its surroundings, the less ordered and more random the system. In other words, entropy increases, delta S greater than 0. So let's apply entropy to living organisms. Living things are highly ordered, and we require constant energy input to maintain ourselves in a state of low entropy, low disorder. Just take a moment to understand this sentence. We know that we are made of atoms, and that those atoms organize into molecules, and that those molecules organize into organelles, and that those organelles organize into cells, and that those cells organize into tissues, and that those tissues organize into organs, which organize into organ systems and living organisms. Does this seem random or disordered to you? Living things are highly, highly ordered, and we require constant energy input to maintain this. This is a low state of entropy, low disorder, low randomness. However, if left alone, the entropy of things gradually increases naturally. It takes work and energy to keep our entropy low, to maintain homeostasis. If we stopped fueling our body, it would stop the cellular processes that do the work to keep low entropy. Low entropy means low disorder and high energy. High entropy means high disorder and low energy. For example, as chemical reactions reach a state of equilibrium, entropy increases. And as molecules at a high concentration in one place diffuse and spread out, entropy also increases. Organisms affect entropy by creating ordered structures from less organized forms of energy and matter, and we call this anabolism. They also replace ordered forms of matter and energy in their surroundings with less ordered forms, and we call this catabolism. In this example, animals consume complex molecules in their food, and they're releasing smaller, lower energy molecules and a lot of heat into the surroundings. Recall from slide 17 that the second law of thermodynamics states that the universe's entropy is always increasing delta S greater than 0. Now you may wonder, if the evolution of more complex organisms, which are highly organized and have lower entropy, if this violates the second law of thermodynamics, and the answer is no. The evolution of more complex organisms does not violate the second law of thermodynamics, but why? Because entropy and disorder may decrease in a particular system, such as a human, as long as the total net entropy of the system and surrounding increases, delta S greater than 0. So we're looking at the net change in entropy, delta S. And just imagine how much catabolism an organism is carrying out on a daily basis to maintain its low entropy. Tons and tons. The organisms may be decreasing entropy to maintain their homeostasis, but it's also carrying out so many catabolic reactions to do so, which is increasing entropy. So again, the net change in entropy is still positive for organisms. Now that you're familiar with entropy, we'll move back to how it relates to metabolism and chemical reactions. We have two types of metabolism: anabolism and catabolism, and those have to do with what we're doing, whether we are building or breaking molecules. When looking at the energetics of the reactions, we also have two types of reactions, and those are spontaneous reactions and non-spontaneous reactions. Spontaneous processes occur without energy input, so there's no ATP required, and they can happen either quickly or slowly. For a process to occur spontaneously, it must increase the entropy of the universe, delta S greater than 0. Processes that decrease entropy are non-spontaneous, so delta S is less than 0. And they will occur only if ATP, energy, is provided. To tell whether a reaction is spontaneous or non-spontaneous, we look at the free energy change, which is delta G, of the reaction. The free energy, also called the Gibbs free energy after the scientist who described it, measures the usable energy. It quantifies energy that can do work. To determine which reactions occur spontaneously and which require energy, scientists need to determine the energy and entropy changes that occur within chemical reactions. This is the change in free energy, delta G. A living system's free energy is energy that can do work when temperature and pressure are uniform, as in a living cell. So, when delta G is less than 0, which is a negative value, the process is spontaneous. When delta G is greater than 0, which is a positive delta G, the reaction is non-spontaneous. It's that simple. When delta G equals 0, the process has reached equilibrium. In our cells, spontaneous processes can be harnessed to perform work. If these concepts and values are new to you, again, just to reiterate, the symbol that looks like a triangle is called delta, and it means change. Delta G is the change of G, free energy, from the beginning to the end of the reaction. The formula is simple. It's G final minus G initial. And this is true of all values that you see with delta. We saw delta S earlier. As a quick example of how to calculate the change, let's use something that we are all familiar with, money. If you begin with $100 and end with $90, what is the change in money value, the delta dollar sign? Final minus initial is $90 minus $100. This equals negative $10. Notice that this is not the value that you still own. You don't owe $10. You do not possess negative $10. That is the change. This is the change in value due to your transaction. You lost money, so you have a negative delta value. Final minus initial. Gibbs free energy is usable energy available to do work. We can calculate the change in free energy in a chemical reaction using this formula. Delta G equals delta H minus T delta S. So what are the components of this reaction? Delta H is enthalpy, the sum of the system's internal energy and the product of its pressure and volume. You can simply think of this as the heat of the reaction. That's all you need to know for this class. H for heat. For endothermic, which means heat absorbing processes, the change of delta H is a positive value because it has gained heat. For exothermic, which means heat releasing processes, delta H is negative because it has lost heat. T is temperature, the absolute temperature of the reaction, and this is measured in a unit called Kelvin. And if you were curious how to calculate Kelvin, it's the degrees in Celsius plus 273. And finally, we have delta S, which is entropy, which is the measure of molecular disorder or randomness. For a biology class, you probably don't need to know how to calculate these equations. That will be saved for you when you hit the general chemistry courses. However, you do need to understand the basic concept behind how these factors interact within a system or a chemical reaction. So let's take the basic example of a flask filled with ice at room temperature. You already know what will happen over time. The room temperature environment will melt the ice and it will become water. And you don't have to add any energy to do this. This is a spontaneous process. So let's look at the values behind this. In this process, delta H is positive because the ice needed to absorb some heat from its surroundings. Delta S is also positive because randomness increased. Molecules frozen in ice are not moving and they're highly structured, while molecules in water are freely moving around. So this was an increase in disorder, so it was an increase in entropy. Overall, this reaction will have a negative delta G, making it a spontaneous reaction as long as there are high enough temperatures to melt the ice. Now, let's begin to apply the concept of Gibbs free energy to living systems. We now know free energy is the usable energy that can cause change and do work. Another way of looking at it is that free energy is a measure of a system's instability, its tendency to change to a more stable state. During a spontaneous change, free energy decreases because we're using it to perform work, and the stability of a system increases. Here's an easy way to remember this. Everything wants to reach equilibrium. If left alone, things will reach equilibrium. When you die, your body reaches equilibrium. It takes energy, lots of energy, for organisms not to reach equilibrium. Remember, homeostasis is not equilibrium. Equilibrium is a state of maximum stability. Have a look at these images. It took energy to create that concentration gradient. Those molecules will spontaneously diffuse toward equilibrium the moment they have the chance. And then what? Nothing. Then no work can be done after that. This biker uses energy to reach the top of the hill, but it takes no energy to coast back down toward equilibrium. At a certain point, the momentum from the potential energy runs out and his bike stops. Then what? Nothing. He's reached equilibrium. A process is spontaneous and can perform work only when it is moving toward equilibrium. Non-spontaneous processes are moving away from equilibrium, and so therefore they require energy. The concept of free energy is also applied to the chemistry of life's processes. Here we have three instances: a man on a high board, molecules clustered together, and a larger molecule. These examples all have one thing in common: they contain high amounts of free energy, so they have a higher G. Because they have more energy, they are less stable and have a greater capacity to do work. The man has high potential energy due to his high position. The cluster of molecules have a high potential energy due to the location because they formed a gradient. And the molecule has high potential energy due to its structure. Below these instances are a new set of instances after spontaneous change and work has been performed. As the free energy decreases, the system becomes more stable. The released free energy was harnessed to do work. The potential energy held within those systems was transformed into kinetic energy. The man dove into the water, or fell into the water. The molecules diffused down their concentration gradient, and the larger molecule was catabolized and became many smaller molecules. In these examples, there is less free energy and a lower G. This means that they are more stable and they're nearer to equilibrium, so they have less capacity to do more work. Remember, a process is spontaneous and can perform work only when it is moving toward equilibrium. So let's apply free energy to metabolism. Understanding which chemical reactions are spontaneous and release free energy is useful for biologists because these are the reactions that can be harnessed to perform work inside the cell. In this image, we see a general pictorial of catabolism and anabolism. Nutrients and carbon substrates are brought into the cell and broken down via catabolic reactions. This creates smaller intermediate molecules. These intermediate molecules are then used as building blocks via anabolic reactions to build larger molecules. Catabolic reactions release energy and are spontaneous. That energy can be harnessed for anabolic reactions. This brings us to another way of categorizing reactions: exergonic and endergonic reactions. But it's very simple. Exergonic reactions release free energy and are spontaneous, while endergonic reactions absorb free energy and are non-spontaneous. Take a look at the red graph on the left, representing an exergonic reaction. On the y-axis is the free energy G. On the x-axis is progress of the reaction over time. A reaction begins with reactants and ends with products, as we know. Here, the reactants begin with more free energy, G. They have a higher G than the products. So where does that energy go? We know from the first law of thermodynamics that energy can't be created or destroyed. It can only be transferred or transformed. Here, the energy is lost to the surrounding, probably as heat. Now look at the change in energy, delta G, for the reaction. To calculate the change is G final minus G initial. And I assigned arbitrary numbers here, 10 and 1, to represent the difference. So it would be 1 minus 10, which equals -9. The delta G would be -9. This is a negative delta G. So energy is released, and this is a spontaneous reaction. Now take a look at the blue graph on the right, representing an endergonic reaction. The reactants begin with less free energy than the products will have. So how does this happen? Obviously, energy needs to be added into this reaction for this to be possible. So let's look at the change in energy delta G for this reaction. G final minus G initial is 10 minus 1, which equals +9. This is a positive delta G, so energy is absorbed for this reaction and is therefore non-spontaneous. Here's another type of graph that is showing the same reactions as the previous slide, but this is called a reaction coordinate, and you will see these types of graphs for chemical reactions such as enzymatic reactions. In the next video, we'll talk about enzymes and dissect these graphs in more detail. Understanding these two types of reactions is important to understanding equilibrium and metabolism. We've already talked about equilibrium. In a system, equilibrium is the condition in which all competing influences are balanced. In terms of free energy, delta G equals 0 in equilibrium. In biology, we use the term homeostasis to describe the steady state of internal, physical, and chemical conditions that are maintained by living systems. But again, equilibrium does not equal homeostasis. Cells are not in equilibrium. They are open systems experiencing a constant flow of materials. Therefore, metabolism is never at equilibrium. Image A is demonstrating an open system using hydroelectricity. It's an open system because there is water flowing in and out. The water flows through the system, spinning the wheel and powering the light bulb. This is a spontaneous reaction as the delta G is negative. Image B shows a multi-step open hydroelectric system, and this better represents our metabolism and our catabolic pathways simply because a catabolic pathway in a cell releases free energy in a series of reactions. Closed systems, or also called isolated systems, are different. Reactions in a closed system eventually reach equilibrium and then they can do nothing. They can do no work. This has to do with chemical equilibrium. Most chemical reactions are actually reversible, so they can proceed in both directions. In one direction, they would be releasing energy into their environment, and the other, they would be absorbing energy from the environment. Look at the picture of the closed hydroelectric system. Reactants within a closed system will undergo chemical reactions in both directions until they reach that state of equilibrium, which has the lowest possible free energy, G, and a state of maximum entropy, S. To push the reactants and products away from the state of equilibrium requires energy. If a cell were a closed system, its chemical reactions would reach equilibrium and it would die because there would be insufficient free energy left to perform the necessary work to maintain life and homeostasis. The cell has three main types of work it performs to maintain life: chemical work, transport work, and mechanical work. Chemical work has to do with chemical reactions. So we have metabolism, such as catabolic and anabolic reactions. Endergonic reactions are pushed by coupling them to exergonic reactions, and we'll learn about this in just a moment. Transport work is what we learned in the last chapter, pumping substances against the direction of spontaneous movement and creating those concentration gradients. The example here is the sodium-potassium pump, which is a form of active transport which requires energy. And mechanical work has to do with movement. So we see contraction of muscle cells, motor proteins such as dynein, which transport vesicles across the microtubules in your cell, and movement of cilia or flagella for cellular movement. So I just mentioned energy coupling, and we've covered the basics of energy and metabolism. Let's do a quick review before going on to energy coupling. There are two types of metabolism: catabolism and anabolism. Catabolism is breaking down molecules, and this releases energy, which makes it exergonic. We express this by a negative delta G. And these reactions are spontaneous and are therefore favorable, and they're moving toward equilibrium. Anabolism is building molecules. This requires energy to be absorbed, which makes this endergonic. and we express this by a positive delta G. These reactions are non-spontaneous and not favorable, and they're moving away from equilibrium. This is a perfect system because we have reactions that require energy and reactions that provide energy. To do work, cells manage their energy resources by energy coupling, which is where they use an exergonic process to drive an endergonic process. And you already know the major energy molecule in the cell is ATP. Most energy coupling in cells is mediated by ATP. These three types of cellular work, which were mechanical, transport, and chemical, are powered by the hydrolysis of ATP. Remember, hydrolysis is using water to break the bond. Hydrolysis, water breaking. Hydrolysis of ATP is very exergonic. It releases a lot of energy. In the cell, the energy from that exergonic reaction, from the ATP hydrolysis, can be used to drive an endergonic reaction which needs energy. And overall, these coupled reactions are still net exergonic. They are still negative delta G, spontaneous and favorable. Let's take a minute to remember the structure of ATP. ATP stands for adenosine triphosphate. ATP is composed of, and you should know this, a ribose sugar, which is also a pentose sugar, ribose sugar, an adenine nitrogenous base, and 3 phosphate groups to make that triphosphate. To release energy, the bonds between the phosphate groups of ATP's tail can be broken by hydrolysis. In particular, energy is released from ATP when the terminal phosphate bond is broken. That's the last phosphate in the chain is cleaved off. This creates ADP, which is adenosine diphosphate, 2 phosphates, and an inorganic phosphate, which is written as PI. And it's called inorganic phosphate because it's no longer attached to a carbon-containing molecule. Because remember, organic just means that something has carbon. So that phosphate group no longer has a carbon in it. ATP drives endergonic reactions by phosphorylation, which is transferring that free phosphate group to another molecule. And the recipient molecule is now a phosphorylated intermediate. Phosphorylation is a very important concept for you to understand. You'll see it over and over again in higher biochemistry and nutrition courses. Here's an example of transport work using phosphorylation. We have the sodium-potassium pump. We know this is a form of active transport, meaning it required energy, and by energy we mean ATP. For ATP energy coupling, ATP is hydrolyzed and that phosphate group attaches to the pump. This activates the pump and creates the phosphorylated intermediate. The carrier protein changes shape to allow the 3 sodium ions to transport to outside the cell. The pump then binds to 2 potassium ions. The phosphate pops off of the pump, which allows it to change shape again, allowing the potassium to be transported into the cell. And the process is then repeated with a new ATP molecule. Here's another example of phosphorylation, but this time with chemical work. Here we're creating an amino acid, glutamine, by reacting glutamic acid with ammonia. Now the delta G for this reaction is positive 3.4 kilocalories per mole, letting us know that this is endergonic and non-spontaneous. So we need to supply it with some energy. Particularly, we're going to use ATP. The delta G for ATP hydrolysis is negative 7.3 kilocalories per mole. Panel B shows the conversion reaction when we couple the glutamine reaction to ATP hydrolysis. Notice the phosphorylated intermediate, that pink triangle, which is glutamic acid, has a yellow phosphate group attached to it. So that is the phosphorylated intermediate. This activates the molecule and allows for the conversion to the amino acid glutamine. ADP and PI are released as products here. To figure out the net G for this coupled reaction, we simply add the delta G's for the two reactions present. So positive 3.4 kilocalories per mole and plus negative 7.3 kilocalories per mole. So our net delta G is negative 3.9 kilocalories per mole. This reaction is net negative delta G, making it spontaneous and favorable. So that is how ATP energy, that exergonic energy, is used to drive these endergonic processes. Energy coupling with ATP is used throughout your body, and you use a lot of it. But luckily, ATP is a renewable energy source for us. We can regenerate it by the addition of that phosphate group back onto that ADP. The energy to phosphorylate ADP comes from catabolic reactions in the cell, and this is called the ATP cycle. The ATP cycle is a revolving door through which energy passes during the transfer from catabolic to anabolic pathways. That wraps up the energy and metabolism portion of this chapter. The next video will focus on part 2 of this chapter, which is enzymes.